Let me close with just a few comments from you, Jorge. Your institution has been very active at looking at these checkpoint inhibitors and combinations in AML. Does this have legs?
Well, I mean, we certainly have very high hopes for that because I think there is the potential for immune approaches in AML. We think that there may be something there. So we've been looking at many different ways of using these checkpoint inhibitors. We have more experience with nivolumab. That's where we started. And there's a few studies, and they're being presented here. One is in combination with azacitidine, first in relapse, refractory relapse, and then some data in the newly diagnosed older patients. And then also the more intensive standard combination, cytarabine and idarubicin. There are single-arm studies. Certainly we've shown the safety, although you do see these toxicities that you expect, these pneumonitis and nephritis and all these things. We have good responses, and we have some comparisons with historical data that make us believe that there is possibly some improvement. It certainly doesn't look like it's a dramatic shift on the curves. There is some trends, and these are historical controls. So we have to take those results with caution, but there may be something. We also have a maintenance study that we showed at EHA that also suggests that these high-risk patients for relapse maybe are maintaining the responses longer. I think we still have a ways to go to understand which inhibitor, how do you combine them, what's the best setting at the beginning when they're in remission. There's a lot to learn. I have the bias to think that they will find their way in leukemia, but I don't think we're there yet. Yeah.
And I think there are a lot of other immune-based approaches that we're taking. Passive immunity, we're not done with antibody-drug conjugates. With the approval of gemtuzumab, I think there is hope that we can improve on that and improve the outcome for our patients. Then the bispecific T-cell engagers and the DARTs that will engage the patient's own immune system. And then, of course, CAR T-cells, which is a panel discussion of all of its own. So I think we better not touch that one.
Yeah.
So I think this has been, extremely informative. Before we end this discussion, I'd like to get closing thoughts from each of our panelists, okay? So let's start with you, Jorge.
Well, I think AML has gotten a lot more exciting from the point of view of the knowledge and the activity, the research, et cetera. But importantly, much more hopeful for our patients because we are starting to see some improvement in outcomes in areas where we didn't have before. There's a lot of room to grow and to improve. I mean, we're not done in any of the areas of the drugs where we have approvals, and of course, there's many areas that are still uncovered. But I think it emphasizes the importance of characterizing well our patients so that we can apply these drugs when they're applicable in individual patients, to use them wisely and know where we have good data and when there's no indications, et cetera. And of course, continue enrolling patients on clinical trials because that's what's led us to these approvals. There are many more tools and many more drugs under development. If we do it well, I think in a few years, the landscape and the outcome of our patients is going to be dramatically better.
Sasha?
I think we spent many years arguing about things like how much daunorubicin we need to give patients, and we're not talking about that anymore because we've got better things to talk about. We have come to a point where we have to say, the questions we have to answer have to do with how do we take this new and exciting drug and make it work the best, and how do we find as many drugs for as many patients? And they're there. There really are some really exciting avenues. This has been a great year in drug development for AML, and I can only hope that 2018, with all the drugs on the landscape, whether it's immunotherapeutics, new FLT3 inhibitors, IDH1 inhibitors, a number of drugs that are coming down the pike. These could be, at our fingertips very soon with drugs that we hope will come approved in the near future. So, it's a great time to be an AML doc, I think.
But you don't really know the right dose of daunorubicin still, do you?
Of course. Now we can argue about the right number of Mylotarg.
Right, exactly.
Well, yeah, we didn't have time for that.
Eunice? So yeah, I'm also incredibly encouraged. I think that, I hate to use this sort of a stock term, but I think we are moving towards a truly personalized approach to AML in individual patients. I think that we have struggled with the complexity and the heterogeneity, both of our patients and the biology of the disease for many years. We're finally beginning to crack that. And what I think is the most exciting thing is not only these novel drugs targeting the immunotherapy immunotherapy and microenvironment coming down the pike. But I also see now that we have known active agents, a lot of the discussion that I've seen at this conference is how to combine. We have one agent, this agent, this agent. Why don't we combine 3 agents? Why don't we take an IDH inhibitor and a BCL2 inhibitor and Mylotarg? Why don't we take liposomal 7 and 3, add a FLT3 inhibitor? So I think the real fun is going to be, as in myeloma, figuring out all the myriad combinations that could be possible over the next few years and seeing which one is really going to be the best. So I'm excited to see that we actually have active agents that we can play with and layering them together and really looking at the patient, looking at the disease, and figuring out which thing to take from each category to make that patient live the longest.
Well, you guys have taken all the thoughts I had, so I'm done, and I think we should leave it at that. Thank you for all of your contributions to this discussion. On behalf of our panel, we thank you for joining us, and we hope you found this OncLive peer exchange to be useful and informative. Thank you.